thank you all very much for joining. I'd like to extend my thanks to those who've come back and haven't been put off by the previous 4 we've had of these. So this is our 5th one tonight. Before I start, I'd just like to sort of say exactly how we're going to run this, which is it's a very informal. We're very fortunate to have Per with us today. We're just going to chat a little bit about Per himself, a little bit about how he got to where he is with his work. Then we're going to discuss his theory and then have some questions around that. By all means, put your questions in the chat. I will try and deal with them as best I can. They're often best if they're a bit more generic rather than specifically for yourself, but any questions, by all means, put them in there. I'd like to thank WPC for setting this up. This was one of the good things about COVID, was this move into a more virtual world. So this is a sort of new venture, and we think it's gone very well, so well that we're actually going to do it again next year. So we're hoping that's going to carry on. Unlike COVID. And I'd like to thank, Supernus Pharmaceuticals, because without them and their support for this, we wouldn't be able to conduct such exercises as this. So thank you very much for supporting this. So without further ado, I'm going to introduce Per, or I'm not going to introduce him, he's going to introduce himself. So Per, lovely to see you. Now, I know that you're a professor in Aarhus. I'd probably pronounce that incorrectly. Now, a lot of people might not even know where Aarhus is. So tell us a little bit about yourself. Tell us how you got into research, where you began life, whether you've always been in Aarhus. A little bit about that, about your background would be very helpful.
Thank you very much, Roger. And thank you very much to the WPC and you, Eli, for inviting me. I'm very honored to be here. So yeah, first of all, it's pronounced Aarhus. In Danish.
I got it wrong.
It's the second biggest city in Denmark. Almost nobody knows it. Everyone only knows Copenhagen. However, I can say that Aarhus is an older city. It was established by Vikings more than 1,000 years ago. So it's actually an old Viking settlement. Anyway, as you said, Roger, my name is Per Borghammer, and I'm a professor in nuclear medicine. And well, how did I become that? I always, when I was a medical student, I was, of course, mainly and almost only interested in the brain because it is the most interesting organ. So at least I thought so, and I still think so. So I always knew that I wanted to do research, and I really wanted to be a neurologist as well. So I started a PhD, and then,
Was all this in Denmark, Per?
All of that was in Denmark, actually still in Aarhus, because there's kind of there's been a lot of neuro research going on in Aarhus for many years, especially in imaging and PET. There's an old kind of well-established PET imaging facility with advanced scans. So I kind of went there and asked if I could do a PhD, and then something happened, and this is a good example of one of those instances in your life where some random thing happens and it changes your life. In the sense that I came there and I talked to the chief, and he said, well, I have my colleague next door. He just received a bunch of money to do a new project. You can go and talk to him. And that bunch of money was for a Parkinson project. And I'd never had at that time any particular interest in Parkinson's. I just thought the whole brain was interesting, but that was Parkinson's. So that became my project, my PhD project. And, to make a long story short, I kind of just ran with that, and I never looked back, because I really think that Parkinson's and, neurodegenerative disorders, and Parkinson's is one of them, Alzheimer's and so on. I really think that, those are I think the most interesting neurological disorders, because I would say they are among the most complicated, if not the most complicated. We simply don't understand them well enough. And secondly, it's also, diseases where we really, really need to get smarter in order to be able to help the patients and develop better treatments. So, yeah.
And what was your PhD on with respect to Parkinson's?
Yeah, it was on Parkinson's, and it was with PET scans, imaging, you looking at the blood flowing through the brain, the glucose metabolism, how is that distributed? Can we use that for diagnostics? Can we use it to learn something about the disease? So I did some studies around that and improved some methodological, some methods to analyze PET, but, in hindsight, it was pretty boring. compared to what I do now.
But you learned a lot. I had exactly the same with my PhD. I think at the end of the day, the only person who read it was me and the examiners, but I learned a lot. So just before we get on to talk a little bit about Parkinson's itself, I thought it might be interesting just to talk a little bit about PET actually, because this is I mean, this is an imaging technique. I mean, most people are used to imaging, obviously MR technique, CT and things, but PET is rather specialized, and it tends to be in rather specialized centers. And in the UK, it's extremely expensive. So, I mean, could you just explain a bit what PET does that other scanning doesn't do, and what its advantages and disadvantages are?
Yeah, sure. Okay, so I think if we start by talking about the usual scan, so if you go to a radiology clinic, they have MRI, CT, ultrasound. These methods are very good at looking at anatomy. You get exquisite images of the size of the brain, and if you have some pathology, a tumor, something like that. So you could say that those images, those techniques are really good at looking at what things look like.
Yeah.
On the other hand, PET is a technique that makes images of function. So what is different there is that we take a molecule, and it could be any molecule, for instance, glucose, sugar. But the point is you then make it radioactive, and then you inject it into the patient. And that kind of sounds dangerous, but it really isn't, because the amount of radioactivity is tiny. So there's really no appreciable risk with it. But the point is, so now you have sugar in your body, but since it's radioactive, we can see where it went. Okay. So if you have a sugar molecule, then you can make an image of the sugar metabolism. If you have a molecule that binds to the dopamine system somewhere, which is very important in Parkinson's, well, then you get an image of the dopamine system. So it is a very powerful technique because you can make images of every conceivable biological process in the body. So that's the upside. The downside is it's expensive. It requires complicated machinery, radiochemists to make these molecules. And sometimes the images are not really nice. They don't look as good as the nice MRI scans, but it tells us something very different about what the biology looks like.
Okay, so that you don't get quite as much definition on structure, but you basically pick up a functional system. So if I want to look at dopamine in the brain, I want to look at noradrenaline in the brain, I can see that with PET, which I would never be able to see with a standard MRI scan or CT scan even. And I suppose just before we move on to talk about Parkinson's itself, I mean, how do you know that what you're looking at is actually what you're looking at? So how do I know that if I squirt in something, it's being taken up by, because we're going to come on to discuss this, how do I know it's taken up by the noradrenaline, the norepinephrine system, or the serotonin system? How do I know that that's what it's going to, rather than it's just sort of binding to areas in the brain which are a bit random?
Yeah, good question. So, and a very important question, but it's actually simple to answer, because you simply have to think about these PET tracers, these PET molecules, in exactly the same way as a pharmaceutical, like some kind of drug you give to patients, which affects some system. So before we are allowed to give a drug to patients, it needs to be rigorously tested in the lab by scientists in tissue samples, in animal models, and so on. It needs to be tested that it's not toxic, dangerous to the patients, severe side effects, all of that stuff. Once you kind of pass that threshold, you have already established with all these techniques that we know that this molecule binds to the target that we are interested in, because all of this stuff goes for the ordinary pharmaceutical industry. And what we do is exactly the same, except that then we just make one step more, and that is how we figure out how to make it radioactive so we can see where it went.
Okay, and just before my final question, I always say it's my final question, and now's my final question. But given that they're binding to molecules and systems that you're interested in, do the drugs that people take for Parkinson's therefore interfere? So if I want to look at the dopamine system and I'm taking dopamine drugs, will that influence the imaging? Or if I'm looking at the 5-HT serotonin system, obviously a lot of antidepressants work on that. So do you have a problem with the drugs interfering with what you're looking at?
Yeah, it's an excellent question, because the short answer is that yes, we need to understand all these things and think about them. So in the case of most people here will know the normal DAT scan, that's used to diagnosing Parkinson's, makes an image of the dopamine system. That has been intensely studied. And we know that ordinary Parkinson drugs like levodopa and so on, the agonists, they don't interfere with the DAT scan molecule. So it doesn't matter if you take it or not. Okay. However, and this is coming more and more, this is becoming more and more important and common, more and more adults and have an ADHD diagnosis.
Okay. And so Alzheimer's and Parkinson's.
Yeah. So it's not uncommon now to see 50, 60-year-old people with an ADHD diagnosis. That was uncommon years ago, but now it's becoming more common. They are treated with basically something that looks like amphetamines. Right. And these drugs, if you take this drug, then your DAT scan becomes pathological. So, and then you say, oh my God, this patient has a severely damaged dopamine system. And then but he's taking this ADHD drug. Then we take that away for a week, scan him again, and then the scan is completely normal.
Okay. So ADHD, you mean attention deficit hyperactivity disorder, not Alzheimer's disease. I got it wrong. Okay.
Absolutely.
Yeah. I was going to say I was getting a bit muddled there. So okay, you said the Ritalin type drugs that people take for these disorders. Okay. So that's very interesting. So obviously PET scanning offers us something which you just can't get with any other imaging, which is obviously in nuclear medicine, and it has some drawbacks, but obviously gives you information which you can't get through any other route. So I mean, one of the questions which arises in Parkinson's, which you have directly taken head on, is this idea that the people with Parkinson's aren't one entity. So I think in years gone by, people sort of said Parkinson's disease, bit of tremor, bit of stiffness, bit of slowness, slowly got worse over time. Everyone's pretty much the same. It'll all follow one course. People might progress a little quicker in some places, but it's basically a single disorder. Whereas people now talk about heterogeneity, different types of Parkinson's disease. So, I mean, in your experience, Per, how do you think we can best conceptualize this? Should we conceptualize varieties of Parkinson's based on clinical features? So we have lots of people with tremor, lots of people without tremor. Should we be looking at people who have problems with blood pressure or whatever? I mean, what's your sort of take on how we can best classify people in terms of heterogeneity, which is the key term, I suppose, people use now?
This is an extremely difficult and a very large question, but I'll try to keep it brief. So I think by now it's very clear to the scientists and clinicians that if you really study, if you really examine each individual Parkinson patient with a lot of different methods, of course, the clinical methods looking at the motor symptoms, but especially all the non-motor symptoms, depression, pain, cognitive problems, problems with the blood pressure, constipation, urinary tract problems, and so on, many others. If we look at this, then it's very clear that some patients have a lot of these symptoms, and other patients have hardly any of these symptoms. They only perhaps have the motor symptoms. Excuse me. And a lot of work, excuse me. A lot of work has been done to basically categorize different kind of subtypes of patients with different patterns of these symptoms. If you follow patients longitudinally, over time, it is also clear that some of these patterns kind of, some types of patients progress faster on both the motor symptoms and the non-motor symptoms, and other patients seem to have many years where nothing much happens. So, and all of what I just said is simply looking at the clinical symptoms. Yeah. Okay. If we do basically the same thing, but try to be really, really thorough and use many different types of scans, blood samples, and so on, maybe biopsies from patients, look at many different things, and, genetics, molecules, and so on, we will see the same thing. We'll see kind of that patients tend to cluster into kind of different entities, if you will. Okay. So, yeah. So what you're saying is that
you can classify people, I suppose, into subtypes based on different parameters. So one could be clinical, one could be on samples, one could be on imaging, and obviously in the ideal world they would overlap. So you say this clinical type, a lot of tremor, not much stiffness, has this imaging profile and these changes on biopsies and blood samples.
Yes.
And I think we're going to come on and chat about that in a minute. So obviously, there's the clinical, and we can take it from that. Now, obviously, the other question, which I think relates to your work, which is where we'll go next, is about this idea that before you ever present, so you talk about non-motor features of Parkinson's disease, and obviously we rely on motor features to make a diagnosis. So even in 2021, the diagnosis is based on the fact, I think clinically you look as though you've got Parkinson's because you've got a bit of stiffness, tremor and whatever. But there's this whole idea that Parkinson's begins years before that, prodromal, premotor, promotor, whatever people want to call it. I mean, what's your take on that? I mean, what would you understand about the sort of prodromal stage of Parkinson's? How do you think that what that looks like? And how does that relate to heterogeneity?
Yes, that's an excellent question. And again, a lot of work is being done on this and has been done for the last couple of decades. I think it's very safe to say by now that some patients, some people with Parkinson's disease, not all of them, but some of them have non-motor symptoms 10 years, sometimes 20 years or more before they get the diagnosis. Some of the more common symptoms are constipation, problems with the urinary tract, with voiding and so on, sleep problems, a decreased sense of smell, and some other problems.
Yeah.
So there's no doubt about that. On
but you don't think everybody has that? So you wouldn't say everyone with Parkinson's has that?
Absolutely not. Yeah,
Okay.
Yeah, and I think, and this is a very important point, because if you kind of open a modern 2021 textbook on Parkinson's, now it's you will see nice schematics which kind of suggest that Parkinson's always starts 20 years maybe before diagnosis, and you get your constipation and your urinary problems, then you get your sleep problems and your sense of smell, and then you get the diagnosis. But that's true for some patients, but probably less than half of them, or let's say maybe half of them. The other half have very few of these symptoms, and some patients have no of these symptoms. They just they start with the Parkinsonism.
Yeah. Okay. So this is very interesting, because I think you're absolutely right. I think there's been a swing from we don't recognize prodromal Parkinson's disease to everyone must have it. And the only reason we don't pick it up is we don't ask penetrating enough questions or persist. But my clinical experience is that there are clearly people who have had a long history of problems of acting out dreams, REM sleep behavior disorder, loss of smell. But there are patients absolutely not, never have, and actually don't ever develop it whilst you're seeing them. So this is a sort of, I suppose this is a sort of tension. So we have people who have prodromal features and then develop Parkinson's disease, and then we have people who just seem to present in their classical way. So this is something you've taken head on, Per, I think, and you sort of combined your enthusiasm and expertise in imaging and your neurology to try and better sort of characterize Parkinson's disease. And I know you've got a couple of slides, so perhaps it might be useful at this point if you just sort of lay out your theory, because you have this idea that Parkinson's disease can begin in two different places, body or brain, and they may be slightly different entities. And so I think you've got a couple of slides. So if we have a look at that and you just walk us through that, that would be terrific.
Right. So can everybody see this?
Yeah, that's great.
Okay, so yeah, I hope it's large enough on your screens. I will first talk about the schematic on the right here. And so here you have what we have termed the body first subtype, if you will, and on the right you have the brain first. So the thinking the thinking is that there's a lot of evidence to suggest that Parkinson's disease can actually start in the nervous system of the gut. That's called the enteric nervous system down here. And there's also a lot of evidence to suggest that this pathology, this is aggregated, this is clumps of protein that starts somewhere.
Yeah.
Once it starts somewhere, it can spread. And it spreads through the neuron pathways, so the neuronal connections. So once in this type, once it starts in the gut nervous system, it spreads via special nerves called the autonomic nervous system. So that means that it spreads to the heart and also to the bottom of the brainstem here. Once it has reached the brainstem, then you could say now it's inside the brain. And then it spreads to the middle of the brainstem, to the top of the brainstem, and then throughout the entire brain at the final stage.
So the initiating event in this disease process is outside of the brain?
Yes, it is. Well, theoretically, yes, that's what we try to prove. And yeah. I believe this is the case, that the initiating event here is in the nervous system of the gut.
Okay.
And then it spreads to the brain. So, and this is very, if it is true, it's very fascinating, because it means, in a way, you could say that in these patients, this is only a secondary brain disorder. Yeah. It's actually a primary, perhaps a gut disorder, if you will, at least in the nervous system of the gut, and then it kind of invades the brain.
Okay. And do you think the vast majority of people have that type of Parkinson's, or is that the minority, only a few?
Well, simplified, I would say it's about 50-50.
50-50, okay.
Yeah, okay. Just about. Okay. We can discuss those numbers, but that's not too far off, in my opinion. Okay, so these patients, if true, now, if the gut becomes diseased as number one, then that would mean that these patients would develop things like symptoms like constipation very early, because this nervous system is involved in gut functions, and if this has become damaged, then you get constipated. Okay. Also, the pathology reaches the heart very early, damages the sympathetic nervous system. That would give you problems with your blood pressure, so-called orthostatic hypotension. When you stand up, you become dizzy and so on. And many of these patients have both constipation and orthostatic hypotension before diagnosis. Okay. And then the next
Can I just to be clear, because it relates to a question, but the pathology which initiates in this site is still centered on this protein alpha-synuclein, which everyone associates with Parkinson's. So it's the same pathology you would expect. It's just in a different place.
Absolutely. Yeah. So what I am suggesting here, it is exactly the same pathology, but this same pathology can start in different places.
Okay.
And in these patients, for some reason that we don't understand, it has a propensity to start in the gut nervous system. But once it starts, it spreads via the available pathways.
Okay.
Okay. So then the next thing that happens is that the middle of the brainstem is affected. And there's some neurons here that are responsible for sleep functions. And once they become damaged, people can develop sleep disorders, especially this well-known REM sleep behavior disorder, where when you sleep in your dream sleep, the healthy condition is to be paralyzed, if you will. You only move your eyes, but in this sleep disorder, you are not paralyzed. You can actually start moving and shouting, talking, and so on. So while you're dreaming, you are acting out your dreams, flailing with your arms, sometimes falling out of bed, and so on. Yeah. And this sleep problem arises from damage to the middle of the brainstem, which is below the dopamine system. So the dopamine system is not yet damaged, and so therefore the patient does not have Parkinson's symptoms and does not have the diagnosis yet. Okay.
So these would be the people who have clear prodromal Parkinson's disease, as we were discussing previously.
Absolutely. So as I said, if you read the textbooks, you see a nice schematic with the constipation, the sleep disorder, and so on. This is the sort of patient that we are talking about, that have had this prodromal phase. Okay. However, on the other hand, and this is kind of the new bit, on the right here we have the so-called brain first subtype. So remember, if it is true that this pathology, these protein clumps, if you will, they have to they start somewhere in a single place. And if that first single place is somewhere inside the brain, and then it spreads from there, then you'll get a completely different progression of symptoms. We think that the pathology first arises in somewhere called the limbic system. Perhaps it actually comes via the nose, mostly in these patients, because the nose is intensely connected to this limbic system.
Okay. So in both cases, it could be something in the environment. One gets to your gut, one gets to your nose, and it triggers pathology at two sites. Okay, yeah.
Sure. Okay, so when it's in the limbic system, the dopamine system talks heavily, if you will, to the limbic system. So if you have pathology in the limbic system, then your dopamine system will become affected very fast.
Yeah.
So in these patients, perhaps the very first symptom will be Parkinson symptoms, motor problems. But then the pathology spreads in the opposite direction.
So can I just ask on that? I mean, if you had pathology in your limbic system first, would you expect anything to be there before the dopamine cells were lost, and the most problems would you expect to change in thinking or memory or mood?
That's an excellent question. But that's not really the case, it seems. Okay. You would have you would think, so are they demented, or do they get depressed or anxiety or something like that? And that's not really the case, it seems.
Okay.
Although we have very few data from such prodrome patients, but it doesn't seem to be the case. And I'll be returning to that later in the conversation but I'll just say it now and then I'll return to it. We have another hypothesis, and that is that the body first on the left here, for reasons that I will return to, we think that these patients have pathology in both sides, both the left and the right side of the brain.
Okay. Also
initially, but the brain first patients probably have most pathology in either the left or the right side. Okay. So if this is true, these patients still have one functioning hemisphere, one functioning brain half, if you will.
Okay.
And that is probably what, this
so it can sort of compensate for any problems.
That you don't see cognitive problems and depression and these things.
Yeah.
But just very fast, okay, so just to end this, then in theory, the pathology spreads in the other direction. So it hits the brainstem. At some point, you will get the REM sleep behavior disorder, but it comes after the Parkinsonism. You can also get constipation and so on. Once the gut nervous system is affected, you can also get blood pressure problems. But all of these things comes after the Parkinsonism in these patients.
Okay. So in theory, if this is right, they would obviously present differently, but ultimately everyone would look the same?
Absolutely. So at the beginning, patients look very different, have different symptoms, but at the end, they look more similar. I'm not claiming that everyone will be exactly the same at the end, because there's also lots of other things to talk about here that makes the different. But we already know from clinical studies, we know this, that if you follow patients for 20 years after diagnosis, then most patients will get more and more symptoms of various sorts.
Okay. And just whilst we're on it, Per, I know we're spending a lot of time on this slide, but there's a couple of questions here. I mean, just so we can clarify, I mean, the enteric nervous system, you say the nerves of the gut, within the gut wall, we have lots of nerves, and those nerve networks then connect directly to the brain or the brain directly connects to them. So there is a direct connection between the brain and the gut, because someone was wondering about how they talk to each other. And then the other question was, when it affects the heart, is it really just the pumping action of the heart? So it's a sort of blood pressure problem, because one of the questions we have is whether it changes the rhythm of the heart, and people could get abnormal rhythms, which isn't necessarily something one would associate with Parkinson's. Abnormal heart rhythms, changes in blood pressure is much more the common. So perhaps you could just clarify that.
Yeah, I mean, to the best of my knowledge, the damage, if you will, to the nervous system of the heart, it is the so-called sympathetic nervous system. It is the system that gets activated if you have to run, or if you get scared and have to run away from a tiger, if you will, all of this stuff. This is the part that gets damaged. So you would think that this would be a serious thing, but luckily, it really isn't. Patients seem to do very well, even though they have lost most of this system. Yeah. Parkinson patients can run; they can run marathons, and at least some of them, and so on. So, even though they have lost this system, so it doesn't really seem to be that much of a problem. Also, epidemiological studies seem to suggest, Parkinson patients, they don't die of heart disease more than anybody else does. Okay. So it doesn't, so it's not dangerous, if you will.
Okay. Okay. So we have this. Your sort of model is that there's a sort of prodromal Parkinson's disease is associated with a body first, gut pathology spreading to the brain, and in contrast, there's a brain-centered disease which may spread peripherally. So that's a nice theory, and you could sort of say clinically that explains why some people have prodromal Parkinson's disease and others don't. But is there any more evidence that you're right? So yeah. What's the data that supports your hypothesis, I suppose?
So I will show that. I will explain the graph on the left here, and then perhaps the next slide, and then we can go back to full screen. I'll just go back if I can, just a second, like this. Okay, right. So these are imaging data. And in the top right here, these are healthy controls. They're in the top right because they are normal on both the sets of imaging data. Down here, you have the brain. This is the dopamine system. So when you're to the right here, you're normal. Here you have the body. This is the images of the heart of this sympathetic system. And again, if you're at the top here, you're normal. So the healthy controls are in the top right corner, normal on both of them. Then you have some blue Parkinson patients here, or rather these guys here, they are prodromal patients. They haven't got the diagnosis yet here, but they have the sleep disorder. And to the left here, we have Parkinson patients, and they also have the sleep disorder. And the point is, these patients, they kind of go through this blue pathway. First, they go down. What does that mean that they seem to go down first? Well, that means that they kind of lose the signal in the heart first. So they go down, the heart signal is gone, but the brain signal is normal. Okay. And then they seem to turn left, and now they start losing the brain signal as a second thing. Okay. On the other hand, we have some
but just so I've got this, this is cross-sectional. So this is taking however many 30 patients rather than one patient I followed 30 times over 10 years.
Yeah, sure, absolutely. These are cross-sectional data. So we are following these patients with repeated scans. Well, so these red patients seem to have the opposite pattern. They seem to go left first. That means that they have lost the dopamine system in the brain first, but they are still up here, so they still have a normal heart. Okay. And then secondly, they seem to turn downwards, if you will, because now they start losing the heart as a second thing. Okay. You asked about cross-sectional data, Roger. So I'll say two things. First of all, we know from other previous imaging studies that if you take Parkinson patients, they have had the diagnosis for 10 years, let's say that. Every single one of them are in this bottom left corner, at least 95% of them. Yeah. We know this. Secondly, these REM sleep patients that we have here, and they have a normal dopamine system. We also know that these patients, when we follow them, they will get the Parkinson diagnosis or dementia with Lewy bodies. And we know that when this happens, they have lost some of the dopamine system. So we know that these patients will go this way, right? Because that has been shown as well. So when you kind of put those jigsaw puzzle pieces together, we know that these patients are, all of them, walking towards this left bottom corner.
Okay.
Should I talk about just briefly?
Yeah. So I suppose one of the strengths of your study is that you've taken people very early in the disease process. So if you just simply took 30 people with well-established, relatively late-stage Parkinson's disease, they would all be in that bottom left-hand corner. You'd say, well, they all look the same. So it's just one disease entity. Okay.
Absolutely. And we are currently doing follow-up repeated scans on these patients. I think we've scanned half of them so far. I don't have the data here. Yeah. but every single one of them is moving in the predicted direction towards the bottom left corner.
Right. Okay.
Yeah.
So did you have another slide, Per, on this, or is it?
I have, and I'll try to make this brief so we can go back to full screen. Okay, so what is this slide about? This slide is about studying brains from deceased Parkinson patients, like the neuropathologists have been doing for many decades. So, and then they look at, so where is the pathology in the brain? Where are this Lewy pathology? What are the patterns? And the point is here, if we compare this left, if we start with this one here, this is kind of a graph of where the pathology is in a type of brains. And the blue box here, this is the brainstem, if you will. So this is where, if the pathology started in the gut, you would expect the pathology to enter here in this blue box.
Because that's where the gut nerves come into the brain.
Yes, and the heart nerves, if you will.
Yeah.
Yeah. So the point here is that you can find kind of very early cases, and they only seem to have pathology here and nowhere else.
Yeah.
And then if we move one step to the right, if I like to call it a skiing slope. It's like a wedge shape. It's high in the left, and then it kind of tapers off here. So you can recognize this same wedge shape here, but this type of patient, we think, are those patients here that have just lived a little longer. So the blue box, that's the entry point. Here now the pathology has become worse, and it has spread upwards in the brain here. And then this is the next stage. You still have the wedge shape. And this is the final stage where you have a lot of pathology in the brainstem, and then it kind of tapers off. But I'm sure everybody can see that this seems to be the same shape progressing to the right. It's kind of a landscape that's just kind of rising upwards everywhere.
So it sort of starts in the brainstem and then it just goes everywhere.
Yeah.
Yeah.
Yeah. But in a kind of a very stereotypical fashion.
Yeah. Yeah.
And of course, my hypothesis is that this type of pattern that you see in brains, in biobanks, this is what we call the body first. This is compatible with the clinical patients we see, as we discussed before, where these patients probably had constipation and so on, because clearly the periphery was sick before, at least that's what we hypothesized, before everything else, because yeah. Okay.
Okay.
And then the second archetype, if you will, that the neuropathologists are describing, this is what I like to call the mountain shape, because the red box here, the pink box, this is inside the brain, if you will. This is that limbic system.
Yeah.
So you can find patients, early patients, that they have a lot of pathology in this limbic system, and then it kind of tapers off in both directions. And if you look at the brainstem here, there's hardly anything. And then the next stage, you still have the mountain shape. Now you start to see, this is a little bit of pathology here in part of that, autonomic system, but nothing down here.
Yeah.
And then the next stage is here, and the next stage is here. But once again, you see it's a profile like a mountain, and then it climbs and climbs and climbs. And this profile is very different from this profile.
And so this would help, sort of, because the top one, many people will be familiar with the Braque staging, and when that was proposed as the model for Parkinson's,
yes,
there were people who said, well, that's true, but it's not true for every case.
Yes.
And your data would reconcile that, because it would say you wouldn't expect it to be in every case, because actually there are two distinct pathological patterns which fit with the two clinical patterns which fit with the imaging patterns.
Absolutely. That's the point. That's kind of putting everything together and making a hypothesis that can explain these data from the biobanks, the imaging data, and the symptoms from the patients.
Okay. So one of the questions we have here, Per, is relates to, which I suspect you won't know the answer to, is why. So the question is, is given that one comes in through the olfactory system, one comes through the gut, obviously you could say, well, what could that be? Is it some toxin? Is it an infectious agent? But the question is it just that, there is a specific infectious agent that gets in at one site and triggers a pathology, or is it that it comes in at both sites, but there's a sort of local immune difference in patients which determines how you handle it? So is there any infection that just triggers something and there's a problem in the patient's immune system at specific sites, or is it different toxins coming in through different routes, or neither?
I think you asked 10 questions in that, in that question, Roger. So I'm sorry if I will be flailing a bit here. But anyway, okay, I think the first thing I'll say is that if everything we just talk about is true, then it is a new way to look at the disease.
Yeah.
And we already know that there's many risk factors that increases the risk of Parkinson's. We know that there can be toxins, like pesticides, heavy metals. There's more and more evidence that if you have repeated infections, viral infections, that can increase your risk. Inflammatory bowel disorders can increase your risk. We know there's many different genes that increase your risk, and so on and so on. So there's many things, mitochondria and so on. But in this new framework, nobody, to my knowledge, has tried to take a new look at what we know about these risk factors in this new framework. Okay. Because it might be, I'll just give you one example. So there was a nice paper last year, 2020, where they had a whole bunch of Parkinson patients, I think like 500. And then they knew a lot about their symptoms. They had asked them about their jobs. Did they have pesticide exposure or not, and so on. And to, briefly, what they showed is that they could basically order the patients into two clinical types. And when I saw this paper, I said, well, yeah, those are the body first types and those are the brain first types, because these guys have REM sleep disorder, they have constipation and everything, and these guys don't really have that. Okay. And the interesting thing is that they also reported in that study that the body first, they had an increased frequency of pesticide exposure.
Okay.
And whereas the others did not. So, I think this is the only study I know of where you could kind of dig this observation out of the data if you know what to look for.
Okay.
But really, this work needs to be started now, I think.
So I suppose the question could be, is it environmental? Is it intrinsic genetic? We don't know, because no one's actually divided the data which has been presented in epidemiological studies around environmental toxins or genetics in this way. They haven't stratified around body first, brain first. It's always been about just Parkinson's disease. Yeah.
Can I say one thing? Just one more thing, Roger, because I am, in my opinion, I think there's a lot of data to suggest that this is complicated, and there's both genetics and there can be environmental factors. So I think both are important, not necessarily in all patients, but definitely in Parkinson's as a whole. So my point here is that we know of many different mutations that increase the risk of Parkinson's. And I'll mention just two. We know there's something called LRRK2, that's one type of mutation. And then there's something called GBA mutations. And, never mind what that is. That's not important right now. But, I have been digging through all the studies I could find about LRRK2 and GBA. And in my opinion, it seems like the LRRK2 patients, they have symptoms that look a lot like the brain first type. They rarely get REM sleep behavior disorder. They don't get a lot of constipation and all that stuff. On the other hand, the GBA patients with the GBA mutation, they seem to look a lot like the body first type. When you image their hearts, you can see the damage to the heart. They have more REM sleep behavior disorder than the average Parkinson patient, and so on. So what does that tell us? That tells us that maybe the GBA mutation interferes with a system which makes it more probable that your Parkinson starts in the gut if you get an infection or some toxins or whatever. On the other hand, the LRRK2 mutation, for reasons that we don't understand, increases kind of the spontaneous risk that one day your Parkinson starts somewhere inside your brain.
So that would bring together that, different, we're exposed to different toxins, infections and such like, but your genetic background, which predisposes you to Parkinson's, triggers a pathology at a particular site because the gene effect there is greater as a result of these variants. Okay, that's very interesting. So there was a question about the locus coeruleus, and its pathology and how that relates to your theory. I suppose because the locus coeruleus, has very diffuse projections, noradrenergic. I mean, the question is about commenting on the lack of pathology in it. What would be your response to that, and whether that invalidates or supports your theory?
Okay, so first of all, we know that the locus coeruleus, this is a very tiny nucleus in the brainstem. It's quite close to the dopamine system. And the neurons in the locus coeruleus are actually, you could call them cousins of the dopamine system, because noradrenaline and dopamine is almost the same molecule. And we've known that for decades, that the locus coeruleus is, you see a lot of Lewy pathology in the locus coeruleus in brain banks of patients. So this is a system that gets severely damaged, probably to the same extent as the dopamine system. Okay, so we know this, there's no doubt about that. Secondly, based on my own imaging data, and there's other groups that have shown the same, those patients with a sleep disorder, the REM sleep disorder, they seem to have more damage to the locus coeruleus. We can image that. Whereas if you take patients that do not have the sleep disorder, in the imaging data so far, they have a locus coeruleus that looks like healthy controls. Okay. And the REM sleep behavior disorder is being caused by some neurons that are very close to the locus coeruleus. So you could say that the locus coeruleus damage that we can measure is kind of a what's it called, a telltale, or I mean, it's a sign that that part of the brain is damaged. So it's no wonder that these patients have the REM sleep behavior disorder.
Okay. So a couple of questions have come up here, Pet. So we've got this theory, and you've got obviously data for it, and you've only presented some of it, and you had a lovely paper in a journal called Brain last year where you really took this apart in a beautiful fashion, where we have body first, brain first. So a couple of the questions come up about, do these carry different prognosis? So is one going to do better than the other? And what about response to therapy? Does it make any difference in terms of how you treat these people with drugs we have now? Would you expect one to do better than the other? I mean, you could argue that body first have other symptoms, so that you obviously have to treat other things. But fundamentally, if I gave L-DOPA to body first, brain first, is there going to be a difference in response?
Yeah. Okay. So you asked about the prognosis. So it is pretty, we know that people with Parkinson's disease that had REM sleep behavior disorder for many years, they tend to have a worse prognosis than people who do not have that. And since, I argue that those people are the body first cases, then yes, there is a difference in prognosis. And secondly, also,
and how big a difference would that be, Pet? So if I was a patient listening to this, should I be really worried if I've got a REM sleep behavior and Parkinson's? Am I going to do dramatically worse than someone who doesn't have that?
Well, not necessarily. Absolutely not, because maybe we could say, so there's so much variation from patient to patient. So it is, I don't think that any, Roger, you are the neurologist, but I don't think that you would ever say to a patient that you could have a crystal ball and how this patient will.
So I suppose what you're saying, if I had 100 people with Parkinson's and a REM sleep behavior disorder and 100 people without, those with the RBD are going to do less well, but on an individual basis, I couldn't really predict.
Yes, exactly. That's my point. So only at a group level, if you have groups of patients, we can see this pattern.
Okay. Yeah. What about treatment response?
Well, again, there are papers suggesting that those with the REM sleep behavior disorder tend to also progress a bit faster on the motor symptoms. And since, those studies, generally, it's expert neurologists that, treat, give the best possible treatment of the motor symptoms. And despite that, they tend to progress a little faster as a group. So once again, that probably suggests that the treatment works less well as the years go by in that group.
But they would still respond to medication, I suppose, is a critical point. It's not
Absolutely.
that one is responsive and one's not responsive.
Absolutely. You know, if they didn't respond, we would be concerned that this is not Parkinson's but something else.
Okay.
So sure, they definitely respond. Yeah.
Okay. So we've obviously explored these two theories. We're sort of slightly running out of time, but I didn't want to go without you telling us about your tantalizing data about asymmetry. So one of the major unresolved issues, I would say, in Parkinson's disease is why do some people present, well, in fact, all patients really with idiopathic Parkinson's disease present with a problem down one side rather than the other. And it's not just because they use one hand more than the other, because it can be as much left as right-handed in right-handed people. So how does your theory speak to that, the asymmetry of the disease? Is there any sort of insights you can give us as to why people might have it more on one side than the other based on your model?
Right. So here I'll show the final slide that I have. Can you see this slide?
Yeah.
Okay, great. I apologize if it's a bit busy, but I'll take you through it. So first we concentrate on the schematic on the left here. And it's important to know something about how the brain is connected. So if we look up here, what I try to show here is that this tiny random part of the brain here, it speaks almost entirely to the same side of the brain. So this is in the left side. So 99% of the connections from this part is to other neurons in the same side of the brain. Only 1% crosses to the other side. Okay, and this is true everywhere in the central nervous system almost. So what does this? Okay, so this is point number one. Point number two is, as we'd already talked about, if it is true that, you know, when Parkinson's disease starts, it starts in a single place and then it spreads. If those two assumptions are correct, well, then if it starts here in the left side of the brain, well, 99% of the spreading options, if you will, are to the same side of the brain. Only 1% would be to the other side of the brain. Okay, so now if we look at this pink box here, this is a schematic of brain first. Okay, so here, this is very, very early. Now we imagine that for some reason, the very first pathology is here, the red circle here in the limbic system, and then it starts spreading. Well, since 99% of the connections are to the same side, in the left side, then the spreading will be to the left, okay? Now, the second image here is a patient that has just received the diagnosis Parkinson's. So the point is now the pathology here in the left side has spread to the dopamine system, and a sufficient number of dopamine cells have been damaged such that the patient now goes to his doctor and with Parkinson's symptoms, but his or her Parkinson's symptoms will be only from this side because of this great deal of asymmetry. Okay, and at the very right here, we see a dopamine scan. This is what I do for a living, among other things. And here we can clearly see that some patients, when we see them and they have just received their diagnosis, they have a very asymmetric scan. So it is mainly one side of the brain where the dopamine system is lost, not in the other side. Okay, so the point here is that the subtype that we call brain first, they have started in one side of the brain. This ends up causing symptoms and damage only in the one side. Okay. Secondly, thirdly, if we move to the body first down here, now there's a small facet here. So here the pathology starts somewhere in the gut. But for some strange reason, nature has done a different wiring of the system. So both the left and the right brain side, if you will, kind of sends nerves to everywhere in the gut. So that means if the pathology actually started in the gut, it will have an almost equal chance of spreading both via the left and the right. So that means that the very first pathology inside the brain is more symmetrical. It's in both the left and the right side. So this is an early case before diagnosis. this middle one is the case at diagnosis, but here you have more symmetrical pathology in both the left and the right side. The patient tends to have more symmetrical Parkinsonism, and when we scan them, we tend to see a more symmetrical scan with both left and right damage.
Okay. And that's been borne out clinically. So people who would have body first prodromal tend to be more symmetric than people who have not.
Yeah.
Okay.
Again, if you go into the literature, there's many studies that have already reported this. What they have reported is that if you have patients who have REM sleep behavior disorder before diagnosis, once they get diagnosed, they tend to be more symmetrical.
Okay.
And we have done the imaging part of that study and showed that when you look at the imaging scans, well, they are also more symmetrical. On the other hand, the patients without REM sleep behavior disorder, they have asymmetric scans and more asymmetric symptoms. Yeah. So, and I think, I think we've been, scientists have been discussing for 100 years what the reason is because behind the Parkinson asymmetry. And this is the hypothesis that comes out of the brain body first hypothesis, that this is the reason for the Parkinson asymmetry. It is, if you say the reason is the wiring of the brain, only 1% crosses, 99% doesn't cross. That's the reason for Parkinson asymmetry.
So we're pretty much out of time, Per, but that's a great place to finish because that's such a sort of exciting area where we can now better understand this type of problem. I mean, we haven't really touched on so many things. I mean, there are lots of questions here. There's lots of data out there about gut being involved before brain in Parkinson's, from pathology studies, from other groups outside of your own. So, I mean, I think it's been a fantastic hour. I mean, this is an exciting new area. One of the great things about Parkinson's is we think we understand it and then we don't understand it. People come along with new theories. It's been fantastic the way you've managed to explain it. I would strongly recommend everyone to read your paper from, it was last year in Brain, I think. I lose track of time these days, but which really took this apart with imaging. Fantastic work. And not only has it helped us explain body first, brain first types of Parkinson's, but also possibly some of the asymmetry that we see. So thank you very much, Per, for giving up your time to explain it so patiently, to me at least. It's been very informative. Thank all of our attendees this afternoon. It's been a great turnout. This will be recorded, so you can come back to it and you can tell your friends to go and listen to it. The next one of these we have on November the 15th. And then we already have them lined up for next year, so every other month. So do join. So thank you all very much for joining us this afternoon. Thank you very much for the WPC for organising it, but thank you particularly, Per, for a fantastic hour presenting your wonderful new theory.